The Stem Cell Review is presented by Celgene Cellular Therapeutics.
Hematopoietic stem cells are special cells in our bone marrow that sustain blood cell production throughout our lifetime. And they can do so because they have unique properties such as self-renewal ability. They can generate daughter cells that are identical in their developmental potential, such as multipotentiality, which means they can generate all different blood cell types, and engraftment ability, which means that they can home into the recipient bone marrow during transplantation and initiate hematopoiesis in the bone marrow. Traditionally, stem cells were first found in the bone marrow, and that's where they reside mainly during adult life. But the stem cells are not generated in the bone marrow because at the time when the stem cells are born, there are no bones. So they are generated at different anatomical sites when the embryo is developing, namely in the vascular endothelium. And we have now found that this endothelium, hemogenic endothelium, we can find it in the embryo, in the dorsal aorta, but also more recently we found that in the placenta can also generate hematopoietic stem cells. And we also know that stem cells can be found in the cord blood at the time of birth, and we're currently investigating whether these are actually just circulating stem cells or whether the stem cells that were generated in the placenta are contributing to this cord blood stem cell pool. In the clinic, you can use bone marrow directly harvested from the bone marrow, or you can mobilize these cells into blood circulation. And these are the sources where you have to find a living bone marrow donor, and it's hard to find a matching bone marrow donor for every patient. So this is why not every patient can be treated. Cord blood is a source that is perhaps more available, but the number of stem cells that can be found in the cord blood is not sufficient to treat an adult patient safely. They can be used to treat pediatric patients, but not very well for adult patients. Well, when we think about the hematopoietic stem cell, they have to be very tightly regulated, because if they're not, you might get leukemia. So this is why there are specific signals from the immediate microenvironment where the stem cell resides that controls whether cells should be self-renewing or differentiating or dying. And when we take the cells out of their niche and put them into culture, we just don't know enough what are the correct signals that would protect the stem cell properties. And this is why we lose the stem cell. They differentiate or they die. We need to either find how can we harvest the stem cells better, that we can get more of them, or how could we put them in culture and expand them. If they can be expanded in their in vivo niche, why cannot we do this in vitro in culture? Or we may need to find ways how to make these cells engraft better. Or what maybe the sort of ultimate dream that many of us have now with the promise of human embryonic stem cells and induced pluripotent stem cells is that we could take a pluripotent cell and make hematopoietic stem cells from scratch in culture. But this is a long-term goal because we need to first know exactly how does this process happen so that we can recapitulate the development of the hematopoietic stem cell in a culture dish. Well, stem cells have been used very successfully to treat many fatal diseases such as leukemia, inherited immune deficiencies, aplastic anemias, and many more. And also more recently, there have been studies that suggest that stem cells can be also used to treat autoimmune diseases. So there are many more applications to come, but currently only about 30 to 50% of the patients are available to find a matching donor. And this makes me think that there's no reason why not all of these patients could be treated. So we have to find better sources for more stem cells so that we can treat every patient. The idea of using or looking at hematopoietic stem cells in the placenta, actually I have to give the credit for our collaborator, Françoise Dieter-Lenlivre in France. And she had done some studies in avian embryos, in birds, and found that they have a similar developmental structure that serves the same function as the placenta, and this is called the allantois. And they found hematopoietic activity in the allantois, and then proceeded to look at this, is there similar activity in the mouse placenta? Placenta harbors a large pool of hematopoietic stem cells during development. They can both be generated in the placenta from this hemogenic endothelium in the placental arteries. But at the same time, there's another vascular niche in the placenta that expands these hematopoietic stem cells without pushing them towards differentiation. So now the placenta can serve as a source for more stem cells, but at the same time, it can teach us how would you make a hematopoietic stem cell and how would you expand the hematopoietic stem cell without pushing them towards differentiation. So now we need to learn what are the key niche cells in the placenta that support the developing hematopoietic stem cell, and what are the key signals that are coming from the niche, and how the cell is reacting to these signals to make the stem cell and expand the stem cell. And then we can try to apply these regulatory mechanisms, hopefully, in the culture dish. The very exciting thing is that we're now at the stage when the basic science research is actually starting to pay off. That we can find about critical regulatory mechanisms, how stem cells are regulated either from the niche or inside the cell. And these studies are coming from zebrafish, from mouse, even from the fruit fly. And we're actually starting to be able to apply this knowledge to the stage that we can get some limited expansion of hematopoietic stem cells in culture, or we can help the stem cells to engraft and survive better when they're transplanted into the patient. So we can already see progress. And but like I said, the ultimate vision for us and maybe others, the ultimate hope would be to start from the pluripotent cell and make a patient-specific hematopoietic stem cell from scratch. But this, I believe, will be a long-term goal.
At Celgene's Cellular Therapeutics, our proprietary placenta-derived stem cells hold the potential to treat a range of serious diseases. Our unique processes for differentiating and producing treatments from this rich source of stem cells will enable us to deliver these therapies on a scale similar to today's biologic remedies. Proud to be at the forefront of this transformation of modern medicine, we are Celgene's Cellular Therapeutics.